Okay, hello and welcome, everyone. Uh, Welcome to the first of this academic year's seminar series. And there's lots of new faces here. I know there's lots of new students, so welcome to Oxford, for all of you who just arrived. I thought I'd give a little bit of an outline of what these series are before introducing our speaker. And so these are seminars we tend to have every Friday in term time. There's a few things outside of term time, outside that pattern, focused broadly on themes around nature, and a very diverse set of themes around that. And afterwards, we always have a drinks reception around the corner for a chance for some more informal conversation as well. And I'm Yadvinder Mali. I'm a professor of ecosystem science here in geography and director of the Levy-Hume Center for Nature Recovery. So if you're a new master student, you'll probably hear me on Monday morning giving a lecture to you. And just to give you a flavor of the types of seminars we have, they're all on our website there, but they're very broad in range. Next week, we've got Tom Oliver from Reading on fixing ourselves to fix nature, inner change as a neglected lever to tackle environmental crises. The week after, we have a film festival, the Conservation Optimism Film Festival of short films around positive conservation stories. We have a week after, making nature markets work in the 21st century. The week after, transformative scenarios for people and nature. Reimagining Bretton Woods by placing nature at the heart of global finance and governance. Understanding human-nature relationships. Loss of large mammals in Malaysia and lessons learned. Eat, Poop and Die, How Animals Make Our World. And that's Joe Roman, the famous whale carbon person, coming over that. And finally, the screaming professor of English literature, The Screaming Sky, The Strange Allure of Swifts. So you can see it's quite a mixture of seminars. So do come along, and they're open to anyone. So invite your friends and colleagues and visitors along as well. So moving on to today, it gives me great pleasure to welcome back Professor Brian Enquist. Brian's a long-standing collaborator and friend. He spent a year on sabbatical here about 7 years ago. I can't believe it was that long ago already. And we've collaborated on lots of things, from transects in the Peruvian Andes to traits in the high Arctic and various other projects as well. But I think Brian's is probably best known for his big picture thinking. His thinking about general theories of ecology and the biosphere, and how can we approximate the complexity of nature into broad underlying principles. But on top of that, having got to know Brian, he's also really fascinated in natural history and detail. He's a great botanist and is fascinated by the intricate details, and this combination of detail and big picture is a unique quality that Brian has. So he's a professor at the University of Arizona and the Department of Ecology and Evolutionary Biology. He did his PhD at the University of New Mexico with James Brown, and was a postdoctoral fellow at the Santa Fe Institute, and he also is a faculty member of the Santa Fe Institute in New Mexico. So welcome back, Brian, and over to you.
Thank you. You want your water? Yeah. Thank you, Yadvinder. And thank you very much for the invitation. And it's good to be back. And thank you for coming. Okay, so let me see if I can get this started. All right, so for my title today, I don't want you to leave disappointed, because I'm not going to present a general theory, but this is towards. And so what I would like to do is focus on what I think is going to be the important components. And I'm going to do my best to try to put them together to show that what we can do. And I hope to also point to the future in terms of how we can collectively work toward a much more general predictive theory than of not only biodiversity change, but also linking that biodiversity change to the functioning of the biosphere. Okay. So one paper that kind of got me along the route of what I'm going to be talking about today was this review paper published, several decades ago by Stuart Chapin and several others, having to do with kind of identifying this problem of, we know biodiversity is changing, and we know biodiversity is going to increasingly be changing, but we want to know what are the functional and the important consequences associated with changing biodiversity on the planet. And if I were to summarize this paper shortly, it would be this graph. And so in this graph, the authors presented a hypothesized set of linkages for how biodiversity, fitting in these various different boxes, then could be linked ultimately to ecosystem and biosphere level processes. And one of the key aspects of the mechanisms by which species can influence ecosystems was by their traits, their attributes in terms of how they make a living, but also how those traits then influence ecosystem level processes. And so you can kind of see that through the various arrows here. But the problem, well, I guess you can think of this as a challenge, is that this is not mathematical. It's not necessarily framed in a way that we can make at least some immediate traction. These are hypothesized interactions in terms of where mechanisms can come in. And so this is also a big challenge in terms of how we make this more kind of practical, and can we build it into a more predictive science of going from individuals and traits ultimately to ecosystems in the biosphere. So this really is a grand challenge. How then do we understand the functional consequences of changing biodiversity? So can we take something like the Amazon basin, climate change, different human land use scenarios, and say something functional in terms of what will happen to the Amazon, or small little plots, different landscapes then within the Amazonian basin? So how do we scale up our measurements from traits and individuals and measures of species richness and biodiversity to communities and ecosystems? And is it possible that we can actually mathematize those arrows and actually develop a little bit more of a quantitative and predictive science of going from traits and biodiversity up to ecosystem level processes? And so what I would like to highlight today is long-running work that we kind of collectively call trait driver theory. And hopefully as I get through a little bit of it, maybe the wording can make sense here. But I want to emphasize a little bit about why I think that this is a basis by which we can begin to integrate a lot of the knowledge that we have in terms of biodiversity and mechanisms in terms of how biodiversity actually influences ecosystem functioning. I'm going to focus on some of the assumptions that we start with, okay? And I'm going to also emphasize several of the predictions. And I'm going to try to go into several recent tests and some new, even unpublished work that we're now grappling with in terms of how we try to push this forward. So this work is really grounded on the long-term observation that the phenotype is tightly linked to the environment. So as we go along environmental gradients, we know that aspects of that biodiversity in terms of the diversity of phenotypes is linked to changes in the environment. So in this case, going from classic work by Humboldt, as we go up mountains, be it in the tropics or in the temperate zone, we know that vegetation changes. The phenotypes and the functioning of vegetation changes. So there's a linkage between the phenotype and the environment. But we should ask ourselves, why is this linkage there, right? Well, we have some understanding, and I'm sure you all do as well, that there is this linkage between how individuals work in the environment and whether or not they're present, whether or not they're rare or dominant, right? So why is change in phenotypic diversity so tightly linked to environmental gradients? So here's one example. So here's going up this tropical gradient in Peru, a gradient that I've worked with Yadvinder, and Yadvinder has worked quite some time on. And I'm showing you elevation going from low elevation all the way at the Amazonian basin, all the way up to tree line. And we're looking at several different traits. And actually, it doesn't matter so much what these different traits are. The different traits are these different colors. It could be nutrient content, structural attributes of leaves. For this point, it doesn't matter. There are these traits that we measure in these different elevations. Some of them increase, some of them decrease, and some of them don't change as much, right? But together, as we move up this tropical mountain, and we can repeat this in any location, traits are changing, some positive, some negative. Why? Do we have a theory to predict this? Rhetorical question, okay? So for today, I'm going to focus on three main components. I'm going to give you an overview of something that we call trait driver theory. I'm going to do my best to provide some tests of some predictions. And I'm also going to set out some novel hypotheses and some novel work that we're doing. Okay. All right, so trait driver theory. Trait driver theory originates actually from a longer string of papers, work done by John Norberg, Simon Levin, Van Savage, several others, Colleen Webb in particular. So there's some theoretical strands out there that made some important initial inroads of what I would call the mathematical language for how we take traits and phenotypes and then combine them to say something about their dynamics on landscapes and how they influence ecosystems. Okay? And so I would refer you to some of these initial papers from some of the initial theoretical threads. Working with Colleen Webb, John Norberg, Van, and several others, we tried to make trait driver theory more integrative and link with many aspects of what we measure in the field. Okay? And the process of integrating these different theoretical threads, in the process, we called this trait driver theory. And so trait driver theory makes a series of analytical predictions, but one has to start with several assumptions, okay? And some of these assumptions I'm agnostic on, but they're useful because it allows us to make predictions. And so community diversity and dynamics of diversity can be best quantified in changes in the sizes of organisms and in the distribution of their traits. So trait driver theory is based on the distribution of sizes and the distribution of their traits. And from those distributions, we can then scale up to make some more quantitative predictions of what their impact on the environment and vice versa is. Okay, so I'm going to try to cartoon this and make some simple assumptions. And the first assumption is actually kind of obvious in a way because it's kind of the root of a lot of biology and kind of physiological ecology. And that is traits covary with each other and they together combine to influence how an organism or a phenotype then performs in a given environment. So here we have a bunch of traits that may interact with each other, right? Those traits then kind of combine in different ways to influence performance, okay? So what is performance? Performance is like a growth rate. It could be a reproductive rate, could be demography, survivorship, okay? Performance then functions, survivorship and reproduction then can combine to influence fitness, okay? So for the purpose of this talk, I'm not going to be talking about fitness per se, but I'm going to be focusing on performance. And there are a lot of different performance metrics that you can measure. That is how well an individual does in a given environment. You can probably think of a lot of different performance metrics. The one that we tend to focus on because it relates to a lot of ecosystem level processes is growth rate, carbon flux, nutrient flux, growth of nutrients, growth of carbon. We call it the growth function. And so this performance function is actually pretty critical because the performance function combines these traits to tell you something about how an individual then performs. Okay? Traits combine to influence how an individual then performs. The second assumption is that this growth function does have a real physical basis, that one can derive these performance functions if you are very clear what your performance metric is. So here I'm showing you kind of a cartoonish version of the growth function. The growth function is a per capita growth rate. And the per capita growth rate then tells you how the biomass growth of individuals with a given trait Z, right, influences their growth. All right, so this is very simplistic. Traits or influences their growth rate, okay? And we can calculate a per capita growth rate of all individuals then with that trait value. Right? And so another aspect of trait driver theory is that it steps back and asks, okay, we want to know then the traits of all these individuals. And if we want to know the traits of all those individuals, the diversity and the different types of traits that you have there is ultimately due to two processes. In any location on the planet, traits that are present there are going to be due to two different functions. Okay, the first one is the trait-based growth function, and the other is the immigration function. So any plot of ground, wherever you are, is going to be the diversity of traits that you have there is going to be influenced by the traits coming in, the immigration function, and then how well those traits do inside your community or assemblage in terms of their growth rate. Right? That determines the dynamics of the traits, okay? The change in traits then over time. Right? So two processes, the growth of traits and then the immigration of traits. Ultimately, those two processes determine the diversity of traits then that you have. Now, you can trick this out mathematically and make those two functions a function of a lot of different things, right? We can make it a function of how those traits interact with each other, right? In a community, there's winners and losers. We can then influence the dynamics of those traits by how many traits there are. We can put in density-dependent functions and things. We can then also put in what the landscape looks like, source sink dynamics. This is just a shorthand for saying that we can make this more complicated, okay? But ultimately, there's two functions, the growth function and the immigration function. Traits are coming in from the outside, and then stuff is happening inside the local landscape or community. Okay, and so this is kind of the basis by which we can bring in other ecological and evolutionary processes. Okay, so the third assumption is that the diversity of these trait values can be characterized by the trait distribution. Right? Now I'm starting with a single trait, but you can make this multidimensional too. So we can think of a given landscape or a given location to have a diversity of trait values, but then those trait values are going to differ in terms of how common they are. So there's going to be maybe one trait value that'll be more common than others. So certain traits will be rare, others are going to be more common, all right? So there's a trait distribution, and it could look like a lot of different distributions, okay? But let's just assume for the moment that it's unimodal and looks nice and symmetrical. Now, because trait driver theory is based on the performance function, and if I focus then on growth rate or mass, then taking our performance function in terms of traits influencing mass, if we integrate then that distribution, that's the total biomass that's present. Okay. Now, trait distributions can vary in their shape. And what comes out of a dynamical version of trait driver theory is that the shape of a trait distribution tells you about the ecological and evolutionary processes that are occurring. So trait driver theory focuses on quantifying trait distributions because the trait distribution, the mean, the variance, the skewness, the kurtosis tells you something very important then about the underlying processes that are happening. So bear with me here. So we can characterize the mean, the variance. Skewness can be positive, it can be negative. Kurtosis can be positive, more peaky, or it can be more negative, wider tails, okay? All of those moments, statistical moments of a distribution, the shape of the distribution tells you something about the underlying dynamics, okay? Okay, so then you can kind of fast forward. If we integrate across this trait distribution, and if we measure individuals in terms of their biomass, then we can say something about the total biomass of the assemblage or the community. At bigger spatial scales, we can say something about the total biomass of the ecosystem of autotrophs or heterotrophs. And if we combine the growth function with the trait distribution, combine those then together, you can say something about the total biomass production, right? We're summing the growth rates across all individuals. And then if you can relate the growth function to nitrogen and phosphorus, you can say something about how we begin to then calculate the fluxes of nutrients, for example. Okay, so just to give you a flavor, without going too many details. Now we make an additional assumption in trait driver theory, and that is that there is this linkage between the genotype and the phenotype and the environment. And that is an assumption of optimality, right? And that is for a given environment, there is a single optimal phenotype that's characterized by a trait value or a set of trait values that maximizes your performance given the constraints of the environment. Okay? So to say that one more time, there is a single optimal phenotype, and this is what we start with because it makes it mathematically tractable, of trait value or set of trait values that maximizes individual performance in that environment, given the constraints of the environment. And that necessitates then this follow-up that is, well, if you change the environment, one then has to change the optimal phenotype. And so a changing environment results in a shift in the trait optimum. Okay? So we can start by making some inroads by, let's assume that the distribution or performances is unimodal. And so each of these different colored curves are all unimodal curves. Think of these curves as different species characterized by one value of a trait, right? Maybe high nitrogen, then medium nitrogen, and low nitrogen, something in their leaves. And so depending on the trait value and depending on the environment, okay, species or individuals then will be kind of located across that environment in different ways. And so let's assume to start, we have a unimodal function. And so if you go to any community, community A or B, different assemblages, different landscapes, you're going to see different traits dominating, okay? Because those then, according to trait driver theory, then are the traits that should be performing best in that environment. Right. So that's all interesting, right? But now let's make this realistic, okay? Can we actually parameterize this to things that we like to measure in the real world? Well, it turns out that there's a separate line of theoretical work, another line of work that I've worked in partially, something called metabolic scaling theory. Right? I'm not going to have time to go into some of the detail, but what metabolic scaling theory does is ask why then do traits and life histories and growth rate and metabolism change with changes in the size of an organism, the body size, as you go from small things to large things. And metabolic scaling theory is important here because we can begin to bring in one important component, one important trait, and that is the size of an organism. And the size of an organism is important because it's probably the single best predictor of a lot of physiological variation, metabolic variation, and life history variation. Doesn't predict everything, but it's a really good correlate. So what metabolic scaling theory says is that a given physiological rate, a given biological rate, is going to be influenced by what we call B naught and then M. M is mass, right? This B naught, I'm going to give you just a quick primer. That's traits. That's where traits goes, all right? So if you measure mass, size of an organism, and then you measure traits that underlie that B naught value, that then tells you can predict a physiological rate. If you know what those traits are and if you know the body size, you can then hopefully predict the physiological rate. Another component is a temperature component. I'm not going to have too much time to talk about this. This can also get complicated, but the expectation with temperature is that there is a temperature response. And that is assuming kind of more of a physical interaction of the kinetics. If you warm things up, biology and chemistry tends to go faster. And I'll tell you just a little bit about that in a little bit. So this is sometimes also known as the metabolic theory of ecology, several papers on this. But what metabolic scaling theory says is that you can take individuals, but we can also translate that to whole ecosystems. So we can take those same equations and we can write them then for total biomass then present within a community and how that relates to ecosystem flux. Now, trait driver theory tells us the B naught value. Metabolic scaling theory tells us what the mass and that little exponent A, how that varies, as well as the temperature component. So we can combine these together. And if we include the mass predictions, metabolism and growth, scales of body size, there's going to be a size distribution. There's going to be inverse relationship between size and number. We're going to have an ecosystem scaling relationship all associated with body size. Oh my gosh, I just covered a lot. I apologize for that. Please take this with a grain of salt. Okay, we have some predictions because I'm going to come back to this because we can integrate this past work with how growth and metabolism scales with size, with how number of individuals scales with body size, and how the productivity of growth scales up to the entire assemblage. Let's put a pin in that, all right? And we'll come back to it because I'm going to show you how we can take those other theoretical components to bring that in. So when we combine metabolic scaling theory with trait driver theory, we get some very interesting predictions. So we know growth rate then should be, of individuals should be associated with the environment and how close you are to the optimum value of the environment. We also know that growth rate should be influenced by traits. We also know that growth rate should be influenced by your size. And if we combine then within trait driver theory, we can predict things like NPP, your net primary productivity. That's going to be equal to this trait normalization term. But also when we integrate across the distributions, we find that there is a variance term, that V. The variance, that how variable your traits are going to have a very interesting effect. You can think of that as the diversity of traits that you have. But then we're going to have an influence of how much biomass, that M tote, you have. And then there's going to be an interesting exponent that comes from the scaling of metabolism of individuals. So showing you this because it's possible to combine the math to scale up to actually have an equation, a simple equation for NPP. Okay? Now, I want to graphically show what this looks like. All right. And so when we started doing this, like, is this true? Does this actually make sense? But the theory was telling us this. If we look at the trait normalization, if you increase your trait value, the size of B naught, your productivity should increase, but it should scale accordingly. But if you look at the variance in your traits associated with growth, and this was interesting, you should see a decrease. Why is that? Why, as we increase the variance, should we see a decrease? But then if we plot the total biomass with the net primary productivity, we should see a sublinear scaling of around 3/5. If you're interested, that's a whole another little story. I'm going to try to explain this graphically. If you want to maximize productivity, right, short term, and if you exclude fossil fuel subsidies of nitrogen and everything, a big goal of agriculture is to minimize diversity, to maximize short-term productivity at the expense of maybe longer-term sustainability. This is a trait distribution that has zero variance and is centered on trait values that do the best for this one cornfield in a given county in Iowa, right? Natural assemblages are not like that, okay? They're not monocultures. All right, we have variance in traits. And what's interesting about trait driver theory is that it says that there is a short-term cost of productivity, but that short-term cost also comes with a long-term benefit. Okay. So we can make trait driver theory more useful by parameterizing it. So we can take the B naught values, and it turns out that there is a literature on how then to combine traits to best predict a performance function like growth rate. And so I'm showing you one performance function that is based on traits, and it's based on carbon accounting and carbon balance. That is, for any individual, the total biomass growth rate is a carbon accounting problem. It has to ultimately map with the ability then of carbon then to be assimilated, allocated, and then ultimately respired. Here are the main traits underlying the carbon accounting of a plant. The carbon use efficiency, the carbon fraction, how much carbon is actually in tissue, the specific leaf area, the area of the leaf divided by mass of the leaf, beta L, that's the leaf mass fraction of the total biomass, the total carbon of the plant, what fraction of the carbon is actually in leaves. The size of the plant, which you can also equate in terms of carbon, and then that exponent is telling you how, for that size and that amount of carbon in a plant, how fast it grows. This isn't magic. This is just carbon accounting. And the math tells you that these traits have to be what they are in terms of understanding the total growth rate or the total amount of carbon gained per plant. Right? It's just accounting. So we have an ability, and you can do this for water, you can do this for nitrogen, but these are the traits then that underlie the carbon, or then you can do the water or the nutrient budgets then of a plant. Okay. So there are some other interesting predictions that come out. Right? So we have some interesting predictions for net primary productivity that I'm going to get to in a little bit, but then we can parameterize it in terms of traits. So we know the traits we can look at, but we have some interesting dynamical predictions that come out. In particular, the shape of the trait distribution can be a signature of past and ongoing change. And I'll give you a cartoon example. Here is our one community with a mean and a variance, but the climate is changing, right? Underneath then these individuals. We go from an old optimum to a new optimum. So a shift in the climate then varies traits on one side of the trait distribution over traits on the other. So we'd expect to see a differential mortality and survivorship difference between where you tend to be located on that initial trait distribution. So we'd expect to see a shift occurring as those individuals with traits closer to the new optimum do better than those further away, right? And maybe with immigration, we can speed then this change. So as we shift then to this new optimum, we then should see the signature of a changing distribution based on the skewness. Because mortality is not instantaneous, some of these individuals in the tail kind of hang on, right, for a little bit until they blink out of the system. So one signature of a changing assemblage or changing biodiversity is a skewness then in the trait distribution. So here's another way, another cartoon of envisioning it. We start in the black distribution there. That's our initial distribution. Now, some traits I showed you initially were positively associated with temperature. As you increase in temperature, we get increases in trait values. Some trait values are negatively related to temperature, right? Now, if you change the temperature, you increase the temperature, you're going to expect differential dynamics of those traits that are positively or negatively correlated with temperature. For those traits that are negatively correlated with temperature, we'd expect to start to see positive skewness. For those traits that are positively correlated with temperature, we'd expect to see negative skewness. So in a warming world, we should detect the signature of changing communities, the changing in the functions based on the shapes then trait distributions. Okay, so how can we test then some of these different predictions? We've tried our best over the years to install a series of forest plots and work in several other forest plots with other people and collaborators and sample many of these different traits then that are associated with these performance functions so that we can parameterize those functions in order to say something about the functional diversity, but also whether or not the trait, if we can calculate the trait distributions and if we can use those trait distributions to scale up to the ecosystem. So in some of these initial studies, we rely on a set kind of forest network where in many of these, we've sampled traits locally, right? And that's actually really important if we sample traits locally. And so here are some of the results, okay? On the top, I'm showing you the change then in the mean trait values, several different trait values, specific leaf area, right? That's the mass of the leaf then divided by the area of the leaf, the wood density, the height of the plant, the leaf area, the seed mass, the percent carbon, nitrogen, phosphorus, and the N to P ratio. And so for the first row, we're looking at the mean. The second is the variance. The third is the skewness. And then the last is the kurtosis, that is how peaky or distributed then the distributions then are. Now, if you look on the left-hand side of panel of the graph, that's latitude. Basically, this is a substitute for temperature, going from temperate boreal and and all the way down to tropical forests. And then we also have elevation. So we looked at plots, then up and down elevations in the tropics and the temperate zone. So one thing that pops out, yet again, another study showing that the means of the trait distributions change. Some traits are positive, some traits are negative. There's also similarity in phenotypic shifts. That is, if we go up a mountain in the tropics, it's very similar to going from the lowland tropics up then to the highest latitudes. So as we go up and down elevational gradients then in the temperate zone or in the tropics, we see similar phenotypic shifts as we go up and down in latitude. So there appears to be a repeatability then in the shifts, some sort of predictability in terms of how the traits then are changing. Now, we also see as we go into colder environments, the variance changes, the variance gets decreased. We also see that some distributions have positive skewness and some distributions have negative skewness. Okay, so some of these are skewed one way, sometimes they're skewed the other way. But then uniformly what we see in terms of kurtosis, if you looked at the dotted line here, that's 0, all those kurtosis values, they tend to be positive, meaning that traits are clustered. Those trait distributions are overly peaky, right? And all of this is consistent then with this optimality expectation, that traits tend to cluster around optimal values. Okay? Another thing that comes out of this is that it's very important to measure traits locally. So when we look at the correlations between the mean community trait distribution value and climate, like temperature or VPD or choose your favorite climate value, when we measure traits locally in the blue, where we have both intra and intraspecific variation, right, versus in red, where we assign trait values from global databases, we see a pretty significant shift in the R-squared, your ability then to relate that trait then to the environment. So measuring traits locally is very important. Okay, so do we see support for these dynamical predictions? We increase temperature, positive relationship with temperature, we expect to see a shift and associated skewness. Same thing with the traits that are negatively related. We should see associated shifts. What I'm showing you is a test of those predictions. So then in orange, those are the few traits that are negatively related. In blue, those are the few traits that are positively related. We have a few traits in gray that are not related to temperature, that show no signal across the gradient. And indeed, we do see the shift then that's associated with increasing temperature in terms of their skewness. That is, when we go and take a snapshot of these communities in terms of their traits, we're seeing then the fingerprint of the dynamics. So the skewness patterns are consistent then with the fingerprint then of a changing, shifting community. And that's also elaborated by, if you look at the rate of change in mean annual temperature at all the different plots, the faster the rate of change, the greater than the skewness. All right, so that's historical changes then in temperature. Okay, we tried to test the productivity predictions in different ways. Up here, I'm showing you an elevation gradient. This is in Colorado, another place that we work at the Rocky Mountain Biological Lab. We measure the instantaneous carbon flux. We can measure pretty close to instantaneous gross primary productivity, GPP. Here, I'm looking at one trait, specific leaf area, at different elevations. These are cases where we measure the traits of every single individual, hundreds of individuals in each of these plots, and done it across elevation, so we can quantify that trait distribution as we go up in elevation. So fluxes, trait values, can we actually use a parameterized value to calculate if we increase the trait normalization, we can calculate this value. Do we see an increase in the flux? But if we control for the variance, do we see a variance signature? That is, the short-term instantaneous productivity should be inversely related to the variance. And let me just stop here and just ask, based on your understanding, why does the instantaneous flux decrease with the variance of the community? Anybody? Yeah, Vinder, you don't get to say anything. Exactly. As we increase the variance away from the optimum, the average performance then decreases, okay? So those communities that have really peaky, very low variance, we should see the highest short-term rate of instantaneous productivity. So when we do that, we control for other covariables. We increase specific leaf area, which is a key component of that B naught value. We do see the positive increase. And we also see the negative increase, sorry, the decrease in flux associated with increasing variance. Okay. Take another data set, maybe one close to home. Here's Rothamsted, right? Dumped a ton of fertilizer on this place from 1843, one of the oldest experiments, fantastic work. We wanted to look at the old school way of harvesting biomass and calculating NPP, right? Annual harvests. We can actually calculate how much biomass is produced. And we can look at the fertilized and non-fertilized plants. We can calculate the mean, variance, skewness, and kurtosis. But we want to look at how diversity influences productivity, right? Big literature on diversity, species richness, productivity, weak signal. In fact, it's kind of negative. It's interesting that it's negative because it's likely related to over-fertilization and so on, but not a strong relationship between diversity and productivity. But if we go through and we calculate the mass-corrected biomass distribution mean specific leaf area, this is our mean, our B naught value. We see, we explain much more variability, pretty respectable R squared, the predicted positive relationship. But then if we then control also for other covariates and look at the community abundance weighted variance, we do see the inverse variance signal. Now, we can try to take this on the road back to Peru, back to this elevation gradient where we work with Yadvinder. We can look at our predictions for how NPP should scale with traits and the distribution of tree sizes, the total biomass. And we can look at this fantastic elevational gradient, which is a wonderful temperature gradient going from tree line all the way then down to the Amazon. And we can measure several of these key functional traits, again in the field, but then we can also utilize other remote sensing techniques that hopefully will enable us to better and more rapidly measure traits than in the field. And so we can then begin to measure several of these same traits, not only from the field, but also from, in this case, the Carnegie Airborne Observatory. Based on hyperspectral and LiDAR data, we can then begin to get a better understanding, not only the biomass, but also several of these other traits associated with these forest plots. A tremendous amount of work. This actually took several years to put together, thanks to Yadvinder's leadership and many others, also based here at the university. Many papers kind of came out of that work. But what we wanted to do is take the trait driver framework and those initial equations relating the growth function and then summing up the growth of all of these individuals. We then use the underlying kind of same performance functions associated with carbon and then scale then those up where we can then go into each of the different pixels associated with these flyovers, where we can calculate not only the height and the biomass associated with each of the different regions and within the plot in the forest itself, but we can incorporate light availability. And so we can incorporate a light kind of extinction dynamics and how then light availability will influence variation in photosynthetic rate. And then we can combine that with the growth functions to get an estimate then of the growth or the productivity that occurs then in each of the different cells using the same framework than showed earlier. And so when we then do that, we can then take this airborne data of LiDAR and hyperspectral, and by then converting that then into real values of biomass as well as trait values, we can then take this variability then in traits and biomass to then calculate ultimately their productivity. And we can compare then our estimate of productivity with many different estimates. So what I'm showing you here are a few different ways, different methods where we can actually estimate the productivity of several of the different plots across the elevation gradient. What I'm also then showing you is that in the black line, that is the real observed data. And the other colored data points then are different ways of going through with different assumptions and estimates of better estimating than the productivity. So the end result is that there's surprising overlap then between the observed and then the predicted then value. And what we can also do is compare the theoretical prediction, okay, coming straight from remote sensing. That is, we can take the remote sensing traits, and we can also take the field-based predictions and compare those two to each other. And so by taking traits from the remote sensing or taking traits from the ground, we actually get very similar estimates of NPP using the trait driver framework. The other curve that I'm showing here, this MODIS estimated NPP, this is another product. It's a remote sensing product to estimate NPP for this similar location, and it doesn't do as well, right? There's some errors, there's some overlap in a few of the sites, but in general, it does definitely different than the more theory-informed kind of predictions. Anyways, so we can then create, right, using then the remote sensing, some maps of spatial variability in NPP, and based on the trait values estimated, as well as our ability then to scale up. Okay. So I would like to then take this last component in just a few minutes here, and then ask and step back. We've kind of come a ways, and I've showed you several different bits of trait driver theory, but we still haven't answered the question, why do traits change along the gradient other than hand waving? Well, those are the traits that do best in that environment because they're optimal. Okay. We still don't necessarily have a theory for why the trait should be changing. So another way of thinking about that is, why does the mean community trait value shift along environmental gradients? Why some positive, why some negative? Well, if we were to unpack that, the argument is that this is an optimality argument. All right, so what do we mean by optimality, especially for evolutionary biologists? So the evolutionary expectation is due to ultimately this phenotype environment matching. And the environment is selecting for specific traits that do best. But what do we mean by that in terms of do best? So ultimately, this has to do with selection for individuals that perform best in that environment. So we can go through and review a little bit of metabolic scaling theory, fluxes, traits, biomass, temperature. So one hypothesis with why we see traits that are shifting along environmental gradients has to do with adaptation to temperature. So if we assume a simple exponential relationship between temperature and a biological rate, as we warm things up, things go faster up into a point, and then the curve then drops down. But if we focus on this initial exponential point, and if we do this and this Boltzmann type plot where we plot the inverse temperature and we calculate the natural log of the rate, we should see these slopes of these lines. That slope of the line we call E. That is a measure of temperature sensitivity. It says as you increase temperature, how fast or how sensitive your biological rate is to a given process. So temperature is expected to have an influence on growth by this baseline expectation of kinetics. You warm things up, it should go faster. Cool things down, it should go slower. But we know that there are a lot of adaptations having to do with physiology. So one way of thinking about this is that if we plot the total amount of biomass in a community versus its NPP or GPP, we should see a positive relationship. But if we look at then a warm set of communities that are hot and cold set of communities are cold, the cold ones for their biomass should be lower because the kinetics say that they move slower. They just produce biomass slower. But we should see a scaling relationship with the mass effect, right? And the difference then has to do with the intercept. So this hypothesis is another way of recasting this direct kinetic control, that there's a direct kinetic effect on plant growth and productivity, right? It's just you warm things up, they go faster. So when we do this inverse temperature plot, inverse of temperature versus the natural log of mass, We fit a line to that. That is a measure of our activation energy or of our temperature sensitivity. And you can compare that against different sites. So that's a prediction. But there's an alternative. And I'm going to do the extreme alternative. And that is, what if there's temperature acclimation or adaptation? That is, what if these traits are actually adaptations to differences in temperature? That is, traits enabling the individual to perform better in the given temperature, given the constraints of kinetics. And you would expect then, as you move along the trait gradient, the frequency then of traits in a cold site versus a warm site should shift. We have warm traits that enable you to perform better in a warm site, and cold traits that enable you to perform better in a cold site. That maybe this negates the effect of kinetics. And the expectation then is that if these traits then are beneficial to negate the effect of kinetics, then you'd expect that the cold growth traits and the warm growth traits along a temperature gradient are going to be related to the shift in traits, right? We're going to see this uniform shift in traits to from cold traits to warm traits. And that then would predict that if we plot the plots from cold sites and the plots from warm sites, they're going to land right on top of each other. If we plot this inverse temperature versus the growth rate, it's going to be a flat line. And what this says is that the individuals growing in cold sites are growing just as fast as the individuals growing in the warm sites, right? They have specific trait adaptations to deal with that. So we have two extreme predictions. And so in the literature, we use these to contrast our expectations. Do we see a slope? And the literature says we should see a biological sensitivity of around 0.65. But if we see temperature acclimation, perfect acclimation, and perfect adaptation, then we should see a slope of 0. So if we look to the Prue gradient, this is inverse temperature. Here is our theoretical expectation from no temperature acclimation or adaptation, the dotted red line. And the individual points, these are individual forest plots, calculating then the net primary productivity, then the growth rate, correcting then for the size, how much biomass you have there. We actually see there is a little bit of a signal, okay, but it's pretty flat. We see pretty remarkably insensitivity of GPP and MPP to the temperature then of the site. Right? Now, let me step back. When we look at all these different traits, each one of these boxes are different traits and how they change across the environment. Some traits don't change, but there are some traits that are positively related. Some are negatively related. We can then combine them to calculate your growth prefactor, your B0, and we know how to combine them together, and we can calculate B0. And if we calculate then B0, what that should then tell us is that if those traits are not shifting in an adaptive way to temperature to compensate growth, we should see an inverse relationship. The alternative is that we see then a flat relationship. So let's use the traits to tell us, do those traits, when we plug them into the growth equation, to actually predict what the mass normalized growth rate should be? Do we see a temperature signal sensitivity, or is it flat? And when we calculate that B0, the combination, that's what you see. So that's entirely consistent with a shift in those traits compensating for the kinetic effect of temperature. So that once you correct for the size, the biomass that's present in the plots, those traits then enable those individuals to grow just as fast in warm sites as they do in cold sites. Okay. So that's one hypothesis, that the observed traits that we see along temperature gradients actually reflect specific selection for temperature acclimation along temperature gradients. Now, we're also working on trying to see if we can test this, but then also take a different approach. And that different approach, instead of starting from a temperature standpoint, steps back even further and asks, can we do this from more first principles based on carbon? And so this next step integrates trait optimization models into trait driver theory. And I'm just going to give you one example. We're going to look at specific leaf area. This is my last example. This is the leaf mass per unit area. So we can take potential variation in leaf mass, divided then by the area of the leaf. This is a measure of investment and leaf longevity, but also your leaf photosynthetic capacity. Now, these models get a little bit more complicated with the biochemistry of photosynthesis and respiration, but we can take something that's known as the P model, and this Wang et al. Recent paper last year. There's some work then that's coming out of Colin Prentice's lab. And we can start with a leaf, but then from more first principles of physiology, we can then, given the constraints of biochemistry, how carbon then ultimately diffuses into a leaf, the efficiency of rubisco, and how various environmental factors such as stomatoconductance, water availability, and temperature influence photosynthesis, we can take the climate of a given location and say, what then are the traits then that maximize your photosynthetic carbon gain in a given environment? So this is a whole kind of like different set of lectures to get into this, but then we can also take another thread of literature that actually predicts how tall vegetation should be. That is, can we take a different approach to predicting how big something is? So these two threads in the literature is predicting, based on the environment, what we'd expect for photosynthetic traits. And there's another thread in the literature saying, well, can we predict the height of vegetation based on the environment? So we can predict size. And if you can predict some of these leaf traits, you could have another way to actually predict what trait value should be in a given environment. So this Kempis et al. Paper, basically, if I put a cartoon of this, the tree height available has to do with how much water is available from the environment, the flow rate of water through individuals, and see that blue line, the red line, they intersect. That is your predicted height of a given location. It's basically a water balance model associated with water moving through plants. Now, these models start to get a little bit more complicated. And so with work from Mikhail Mustry then at the University of Arizona, we're trying then to take what we know about the scaling of basal metabolism with size, variation in precipitation, variation in air temperature and vapor pressure deficit. And can we look at these plant traits and plant size and then perform an optimization? Given the local climate, temperature, water availability, can we find the traits that maximize carbon gain using the Prentice et al. And the P model approach? And then with the Kempis et al. Approach, can we then also optimize and find the combination of leaf traits and plant sizes that maximize the total carbon plant gain given the constraint of the environment? Now, math is a little bit more difficult, but you can solve this. And for a given temperature, water availability, as well as variability in those, we can then pop out a series of environment-specific performance functions that tells you then how all these different traits then combine to then influence the plant performance in that environment. And another thing that you can do by combining biomass assimilation, water limitation, variation in leaf senescence, and the size then of the individual is you can find the combination then of traits that maximize the growth rate in a given environment. And when you do that, I'll show you one example. We move along local annual precipitation, going from dry places to wet places, going from cold places to warm places. We can actually predict certain traits. I'm showing you leaf mass per unit area because it comes out as very important. Now it pops out a prediction that trait LMA, leaf mass per unit area, is inversely proportional to temperature. And that's what we see repeatedly across our various different data sets, that is indeed the case, as well as the case that LMA tends to be negatively related to precipitation. Okay? Now, you can also look in terms of the sizes and other traits in these larger kind of predictive graphs, but I'm not going to go into this because this is kind of the next steps. But the point is that we now have theoretical bases by which not only to understand the shapes of trait distributions, but also begin to make predictions about what the mean value should be and why traits then should shift along environmental gradients. And we're even at the point where we may have competing hypotheses for the underlying drivers for why these traits then should be changing. Okay, so with that, I just want to end. All right, so I gave you an overview, many cartoons actually, of trait driver theory for how trait distributions of assemblages are ultimately linked to ecosystem level processes. And then we can link then the shape then of these trait distributions to underlying ecological and evolutionary dynamics. So things like the mean, the variance, the skewness, the kurtosis actually relate, to important processes. So these higher moments, right, skewness and kurtosis actually reflect these dynamical processes, right? So this theory extends, I think, the predictive ability of us as physiologists and ecologists. I'm not saying this does everything, it doesn't, and there's a lot of issues still. But what I've been more impressed with is that we can keep kind of pushing this in new directions and making some novel predictions where we didn't have predictions before. And I really do think that this opens up the possibilities to have more of a predictive theory then for understanding changes in biodiversity in terms of their traits, in terms of their functions, and how then that influences ecosystem functioning, right? And I think we're even at the point where we can begin to start to be a little bit more brave and make some predictions about what will happen in the future under different changes in VPD, different changes in temperature in terms of expected trait shifts and functional changes. All right, so I started with that diagram by Stuart Chapin et al. And I think we've actually come a long way. We can begin to map out a lot of those different arrows linking biodiversity to ecosystem level processes then via traits. And if there's just one more thing that I just want to say, I just want to just bear with me. This is the exact last thing I want to say. Has to do with trait-based ecology. Trait-based ecology, and I've been a part of this, I've contributed to the fact that it's still largely correlative. We're still looking at the traits across the environment, and that's not a negative thing. We need to do that. But the next step really is to focus on taking our knowledge and developing more of deeper understanding, okay? And I've emphasized some things like performance functions. We need to know much more about how the performance of individuals are linked to traits and how we can develop theory then for that. I didn't have time to talk about this, but there are some really exciting developments if you study fisheries or if you go into the people that do plankton work in the oceans that are also developing these really predictive mechanistic models based on performance functions as well and traits. But the last thing is that we need competing theory with alternative hypotheses, okay? Because I think ultimately that's how we advance of all of this work. And so with that, I just want to say thank you. And I want to acknowledge several people that contributed to a lot of this work, as well as several different funding sources. So thank you.
Thank you, Brian. Amazing overview and seeing all the latest progress. First thing I want to say to the students out there, this is your first lecture in Oxford. Don't worry, this isn't on the exams. But I hope you enjoy getting this big picture vision. We'll open up to questions now. Just to kick off, I'm wondering what you have there, if I understood it, is a theory of snapshot. If you look at the current distribution of traits, how they are. The skewness tells you that they're responding to warming, etc. What are the steps of turning that into a dynamic predictive theory? That skewness to some extent reflects a fast responding and slow responding species and the rates of change. And how do we make this theory one that
more operational in terms of
responding in time?
Responding in time, yeah. You would need to know something about the timescales at which the organisms are responding, right? So you need to know something about their lifespans relative to the pace of the change that they're seeing. So you're going to have long-lived organisms versus short-lived organisms. So that's probably the first thing I would point to. But then also you would need to know something about how new traits are introduced then into wherever you're interested in. And so you need to know something about the landscape and the interconnections now of the distance between patches that may support, some traits that are not present in a given location. So you need to know something about the interconnectedness then of the landscape to say something about that immigration function. And so that immigration function is actually exceptionally important because that on the timescales that we're talking about is the easiest way to increase the potential variability, which influences the ability then of the assemblage or the community to respond to change, right? So yeah, definitely something more about understanding what shapes the distribution of diversity of traits across the landscape. Yeah, so just quick ideas. Yeah.
Okay. It's for the online audience.
Thanks, fascinating talk. So I get the organisms will have optimal trait values for a given environment.
Yeah.
My question is about what about the environmental variability? So it's the difference between weather as experienced by a plant on a given year and long-term climate. What does trait driver theory have to say about that?
So I just want to restate the question a little bit differently. So you're basically asking, well, what kind of what sets the variance, right? And what about the variability of the climate, right?
Exactly. How do the trait distributions respond to environmental variability in climate, temperature?
Yeah. So part of the answer to that is having to do with the fact that the environment is varying. In fact, one thing we have done several additional bits of work, and we're trying to still parse out the data. But it looks as if you compare environments that are more stable, low variance than in weather, right, versus ones that's highly variance, that variability then in your climate then is actually very important for influencing the variance, right? So in a perfectly stable environment, no change at all, and no good years and bad years, that trait distribution should almost collapse to be dominated by the most optimal phenotype. If you believe all of this stuff, I mean, literally, it should collapse to just one phenotypic value. So one way to keep the variance then in the system is that the climate is variable, right? So in some years, one tail of the trait distribution does well, other years it doesn't, right? So that the variance of the environment is actually really important in helping to keep then that variance. But then the variance is also influenced, the trait variance is influenced by traits coming in, from outside of your given study area, right? So the surrounding landscape, how heterogeneous is the surrounding landscape? So heterogeneity in time and heterogeneity in space. I mean, this is kind of related to also the coexistence theory and so on, so the mystery of how you can keep all this diversity, So yeah, I think that answered your question somewhat.
Yeah, thanks. I just wondered if anyone's tested that.
Has anyone tested that? Kind of. We have looked at, actually, we've looked at phytoplankton, and I believe there's a few other long-term animal data sets where we have looked at, you can compare like different lakes and different environments, one that is highly variable, and there's like pH and temperature, and others that are more stable. And you can then also take time series and look at different points in the time series that are more variable and not. And then ask, in those times of high variability in the environment, what is the variance like? And you get this very nice functional relationship between the local variance that you see and the amount of past variability then in the climate then that you see. Yeah. And you can experiment, with that, by the way. Yeah.
Thanks. It's a related. Thanks. A related question on the terminology. You're calling it trait driver theory, but it seems to me that the traits are often responding.
Yeah.
I mean, would it be better to call it something to do with trait indicator hypothesis or similar?
Yeah, there's a little bit of a naming historical legacy here. But the idea is that there are underlying drivers then of traits, right? Be that the performance function and the immigration function, but also climate and how then that influences it, right? But there's these underlying drivers in terms of what influences then the traits that are available and their dynamics, right? So for lack of a better word, and maybe in retrospect, maybe we named it something better, but good point. Yeah.
Thank you. Hi, Brian. Yeah, really interesting talk. Unfortunately, this is a question of why don't you have X, Y, and Z in the model?
Yeah.
And that everybody gets those. But sort of thinking about the work, for example, by Mark Veland, you've covered what he would call ecological selection and dispersal. So have you thought about adding in sort of the other half, drift and speciation, how that would
You
work?
Definitely can take this. Yeah. So the question is, if you add in kind of processes, evolutionary processes that also contribute to the variance, right, over
Yeah,
time,
and
right?
drift, yeah.
Yeah. Oh, definitely. I mean, it mathematically, it's all the same machinery, but then you can start kind of adding in these longer time scales in terms of what influences the variability then that you see.
Okay.
And yeah, you'd expect then that landscapes that maybe have had higher rates of net diversification that are more heterogeneous would then contribute then to increased variance kind of locally, especially if they have a high kind of immigration function.
Few questions online.
Mhm.
Relay. One's from Matt Disney at UCL.
Yeah.
A lot of this is underpinned, it seems, on the assumptions of optimality traits and
Yep.
relationships. How well does this hold, and have the implications for how it applies under more rapid change been tested? And what are the implications of that?
Yeah, so I think this also relates to the earlier question about, if rates of change then increase, right? Then what does this say about that? And that's a really good question. So ultimately, the ability then of phenotypes to keep up with the pace then of climate change has to do with the underlying biological processes of growth and reproduction, recruitment, and then surviving then to reproduce again, right? And so depending on the time scale of those biological rates to the time scale of the rate of change, right? If the time scale of the biological processes are faster than the time scale of biological change, you'd expect then things to keep up. But when there starts to be a mismatch, then that's when biology gets left behind, right? So
Okay, so a few more
good
online.
question,
And from Jessica, are there any mathematical efforts to account for chromosomal variance traits?
like what? Chromosomal variance traits, like Floydie level, or
I didn't know more detail there, but I imagine.
so okay, so changes in genome size and ploidy level influences cell size, and variance in cell size can also influence leaf thickness. And so yes, there are some connections, I think, with leaf mass per unit area and chromosome content and ploidy level. Do have a few students actually interested in this and working on this. There seems to be some signals there, but I think it's kind of an open question of the connections to that, but really cool.
I'll take one more question from Laura here as well. What about bimodal distributions of traits?
Yeah, bimodal distributions of traits, which you can clearly get, and we've seen them, they exist. There are multimodal distributions. If you step back, they tend to be unimodal. There are different tests. We can test multimodality and things. You can get bimodal distributions of traits in many different ways. If there are multiple optima, for example, so you may expect multiple optima if there's selection then for maybe shallow rooted and deep rooted, because resources may differ, right? So we can have water coming in maybe in the summertime in short pulses versus long-term water availability during the wintertime, deeper rooted. So maybe you can have systems where resources are coming in at different ways, so you can partition resources that way. You can get bimodality if you have two biased immigration functions where you have traits coming in from one side and traits coming in from another that are faster than the rate at which traits are sorted in the community, so they kind of build up on the tails. Yeah, that's another way. So yeah, it's a good question. If I could say, I was expecting to see more multimodality, especially when we start sampling more and more individuals. In general, I'm surprised more with how unimodal they are than multimodal.
Okay,
Yep.
Well, thank you for a talk. Really inspiring discussion. And I noticed that you had a talk about biodiversity and ecology without mentioning once, or maybe once, species. So are species going in the way or in the way of theory and simplification because complexity of species and local and trait? So that's
So what
maybe
about
you will
species,
comment.
right? Yeah. Okay. Where does species fit into all of it? Yeah.
Exactly.
Yeah,
And the
yeah.
other thing was about the bimodality is the hierarchy of trade.
Yeah.
So because you will have trade that will influence your growth rate, biomass productivity, and depending how they are hierarchized, they will affect others. So
Mhm.
they are not independent of each other. And then you would have,
I didn't quite get the last question, but
so the trade are hierarchic, their influence on grow rate or biomass. And because these traits are not independent of each other, they have covariance.
yeah, and that's
And
right.
that will create some bimodal and different optimum, probably. So I was wondering if that.
Can I just take the last question first? So what about trait covariance and things? So the theory then tells you what the covariance then should be, right? So the performance function, if you have a derivation of that based on accounting, then it tells you which traits are in the numerator, which are on the denominator, and then how then they're going to given for a given growth rate, how then they're going to be related and potentially covary then with each other, right? So the theory actually tells you something about the nature then of the covariance structure, right? Which I actually think is really interesting because we actually have some predictions for what the covariance matrix then should be for the traits. But then again, if you have traits then that are unrelated to a given performance function, you shouldn't see, but they could be through other traits, right? And it brings up this whole thorny issue of how phenotypes are integrated and everything, which is tremendously fascinating. And I think we're just kind of scratching the surface, with that. And then the first question was, yeah, species. Oh, yeah, you have to be that person who asked that question, right? So, yeah. Well, look, I'm a species ecologist. I identify species, but what's really interesting, and I'll just take a step back and be just agnostic. If you want to develop then these predictions on these timescales, you don't need species. All right, what you need are the traits of individuals. Now, what's very interesting is that species then tend to be characteristic. They're bags of individuals that have certain trait values and certain trait distributions. So at a given spatial and temporal scale, if you step back, those distributions are species distributions then as well, right? And so they're kind of like more of an averaged, but that's the evolutionary dynamic, right? Of individuals at different trait values, and you get ultimately, bifurcations and speciation, and you get then, trait modes and differences, right? And so you can look at these distributions in terms of traits, but ultimately for a lot of the things that we want to do, we want to know what the traits of individuals are. Now, if you want to do the evolutionary biology, of course you need to know species, right? Just a lot of these questions yet we haven't kind of gotten back there, and that's not to negate the need for importance of species or anything like that. But yeah, yeah,
And I guess you focus on productivity and biomass.
yeah.
What a range of performances like reproduction and things, and the alignment with species becomes stronger.
Yeah, if you begin to bring in more of these different performance functions and start, thinking about fitness, and then yeah, you're gonna start having more of these intersections and things. Yeah.
Okay, so if no more questions, thank you once again, Brian.
Great, thanks for your time.
And thank you, everyone online. And next week, a very different talk on inner change as a driver for environmental change. But the drinks around the corner if anybody wants to join us. So. That's terrific.